Well, thanks again for inviting me to sunny Seattle. It is coming out. Um, I think, we all have big visions of wanting to change the standard of care in brain tumors. We also have little visions of improving our infrastructure, and, this is what we aspire to do, but we're right now still at the basics of the hospital development in Santa Monica. But it's a nice vision. In brain tumor too, we have a vision. We want to do big things. We want to cure brain cancers, and it really takes time and effort, and you need a team. And I always, in the last 20 years, I learned that you need a team of physicians, scientists, clinicians, patients, and I think the real need for our field is not only to understand brain tumors better, but also to develop new drugs. That's really one of the things that we're missing, and to do this as a team and accelerate with multiple institutions working together, which we're getting better and better at over time, as evidenced by some of the earlier talks and later talks. So what we've done is built a small team in Santa Monica focused around brain tumors, translational research, and really trying to change the way we do clinical trials and find disruptive treatments that are really going to help patients. And that's really what we focused on. As well, we have a team that does a lot of quality and outcomes research, because one of the things with brain tumors and many cancers is some of the drugs cause toxicity, and how do we manage the toxicity, improve the quality of life, and address all the issues surrounding cancer in these patients. So our basic goal is to accelerate biological understanding of these diseases, drug discovery, and to help do faster clinical trials. And this is because cancer is not that simple, and in particular brain tumors, and you've heard some of the information, that it's a very complex disease with a lot of factors going into it, including the genetics that we discussed, but all the signaling molecules that are deranged and how it affects the biology of the tumor. And this is a Hanahan-Weinberg chart that talks about the various issues, including cell death, angiogenesis, genomic instability, et cetera, that are some of the factors that make cancer so difficult to treat. We usually tend to focus on one factor at a time, and what we really need to do is integrate all that information and design better treatments that address everything, or combinations. And that was the talk that you just heard about before. And that's what we're trying to do, because when you look at a tumor, you'll see it's got all kinds of, besides the tumor cells, there's a lot of immune cells in there. And we don't really understand what they're doing, how they're promoting the disease, and how we can utilize drugs to affect the immune system to kill cancer. And that's really where the next wave of genomics, immunotherapeutics, and informatics is going to, that we're all trying to do right now. And so we're trying to do this by getting tissue, as you heard about, getting tumor tissue, getting blood, getting spinal fluid, and trying to understand what's in there, and can it be of value in telling us something about the patient where we can act upon in terms of treatment in the future. But it's hard to do, and I'm going to go through some of these complexities and at least some of the things that people are doing and we're doing to try to address that, and something that we can talk about at the end as well. Tumor heterogeneity, you heard about this, and it's a real thing, and we see it all the time. But, and here's examples of this heterogeneity. This is a patient that recurred after several surgeries, and several clinical trials failed, and this was early 2000s, where we just tried Gleevec, one of the first targeted therapeutics approved for cancer. And you can see he came back surprisingly a month later with this dramatic shrinkage of the tumor. And that's really what that's the hope of targeted therapy, is to try to figure out why did this patient respond, and is there a marker, and can we select patients better? And it turned out the patient had amplification of PDGFR alpha. We then started a clinical trial in the last 6 years where we select patients and give a newer version of Gleevec called nilotinib. And we've had some really good responses, and the trial just ended last year, and we're still analyzing the data, but the basic point is we can do a better job if we take a drug where we know the mechanism of action and actually select patients. And that's what targeted therapy is all about. That's what precision medicine is all about. And these are the types of responses we hope to get at a higher frequency than we're getting right now. But when we don't have trials, what do? We talked earlier about how we are limited in terms of timing of information, of getting data about the markers and how we can utilize that in the clinic. There are many barriers, including getting that information quickly enough, and then also being able to get those drugs, especially since it's off-label use. But we have been successful at doing it in certain cases, and here's examples of the complexity of how the tumor actually changes over time. So this was a patient with glioblastoma, unmethylated, so very poor prognosis, and the patient progressed after chemotherapy and radiation. But we knew the patient had EGFR and mTOR activation mutation, and we started the patient on a combination of 2 drugs that target that pathway, Afinitor and Tarceva. The patient had a good response for over a year, and then the tumor came back. It progressed. And at the time, we took the tumor out again, and the mutation was gone. So this is tumor evolutions under selective pressures. But the patient, for an unmethylated glioblastoma, survived almost 3 years, which shows that there is value to this approach. It's just a N of 1 individualized approach, and we have to get tissue. We have to understand what changed in the meantime in order to be able to treat that specific patient. And that's really the challenge, is getting an infrastructure to do this on every single patient and treat them in different ways and different drugs. It's quite a challenge. And you heard we can't operate on all the patients, so these are selected cases where we can actually, reasonable to go in and back in and get tissue out. Here's another example of a patient with that, again, progressed after radiation chemo, but interestingly, he had a BRAF mutation. This is a mutation originally discovered in melanoma. So this is a glioblastoma with a melanoma mutation. And we treated this patient with Zelboraf, which is a BRAF inhibitor, oral drug, and he responded for well over 2 years, over 2 years, still responding. So again, example of precision medicine in action, and this mutation is not found in very many patients, so this was a unique finding. Another example of a different mutation in a pediatric patient with a brainstem high-grade glioma glioblastoma, again, failed a whole bunch of standard therapies, including Avastin, including immunotherapy. We found a PI3 kinase mutation, which predicted sensitivity to a drug called everolimus, which is a breast cancer drug. It actually gets into the brain reasonably well as well. And this patient responded dramatically with almost complete resolution of the lesion and still alive 2.5 years later, just on single agent everolimus. So again, really, we can help individual patients if we understand the genomics, what's driving the cancer, and picking drugs, if there are drugs available for that mutation. And so the idea of doing, I think what Charles alluded to earlier, about doing more personalized precision trials of understanding the mutational profile and then placing patients in buckets of what's driving their cancer. Is it EGFR? Is it mTOR, PDGFR, et cetera? And doing that. And we're also thinking about this in terms of immunotherapy as well. What are the immune markers, and which immune drugs can also predict response to each patient? And I'll talk about that in a bit. But this takes a lot of effort and a team-based approach to do this. The other issue is really, that's the heterogeneity. The other issue is making sure the drugs get into the brain, the blood-brain barrier, which is still a challenge and not much talked about. But this is one of our biggest problems that we have in the field. And many of the drugs don't get in very well. Temodar actually gets in reasonably well, 20% blood plasma ratio, but most of the drugs are less than 1%. So that's one of the challenges that we have. And there are several companies trying to make better drugs that get into the brain. And here's an example of one of the companies that we worked with that developed Taxol, which is a chemotherapy breast cancer. It doesn't get into the brain, but they modified it so that it does get into the brain through a specific transporter. And so when you do get Taxol into the brain, it actually has reasonable activity, as you can see here in this one case where a patient before treatment, after treatment, had a partial response to the drug. So again, we need to understand blood-brain barrier delivery and how we can overcome the blood-brain barrier with old drugs as well as new drugs. But this is the biggest thing, and I think something that we're all thinking about these days is the immune system. We've heard amazing things from the melanoma world, lung cancer now, bladder cancer, and other cancers where these immune-stimulating drugs are really curing patients like never before. Melanoma was what brain tumors are right now. Back 5 years ago, where everyone, the prognosis, we weren't talking 5-year survivals. And now it's over 50% 5-year survival because of these amazing drugs that are really saving lives. And how do we incorporate that into our world in the brain tumors? We have done a lot of immune therapy studies over the years, over the last decade or so, from antibodies at the top, EGFR, PDGFR, VEGF, to vaccines made from peptides or from the actual tumor cells, viruses, oncolytics, small molecule inhibitors, and more recently these checkpoint inhibitors, Opdivo, Keytruda, and also now CAR T cells, engineering your own immune cells to attack specific targets which are present on the tumor cells. And there are interesting, each of them, there's interesting data of patients responding, small numbers of patients responding, but nothing amazing enough, unfortunately, that anything is approved at this point, but nonetheless interesting. One of the difficulties has been that we've tried all of them. You need to get rid of the tumor because, unfortunately, we need to give steroids to our patients because of the swelling that occurs with brain tumors, and so most of these studies are done when there's near total or very minimal disease, and patients don't require much or zero steroids, and that's been the real issue. And the other side, flip side, is when we give these immunotherapies, especially checkpoint inhibitors, we get tremendous inflammation in the brain and toxicity and the need for steroids. So we're doing a yin and yang of trying to maximize immune response, but also managing the side effects of our treatments. And that's really the challenge in the brain, is to how do we manage all that. But there are opportunities again, and I'll show you a few examples of our own cases and in the literature where you can get a great response in the brain without toxicities. This is a patient of mine that has a rare type of brain tumor even rarer, called primary CNS lymphoma. This is a lymphoma that only occurs in the brain. And this patient had multiple rounds of chemo for 6 years, multiple rounds of chemotherapies, immune therapies, et cetera, and then eventually radiation because we didn't have any other options. And anyway, the tumor came back right away after radiation in multiple locations. And we started the checkpoint inhibitor called nivolumab at the time, which hadn't been approved for the systemic lymphoma, but we had it as a compassionate use. And amazingly, the tumor responded within a month, and he's 2.5 years out, still doing amazingly well after immunotherapy. He's got some immune side effects, but those are manageable. But again, in principle, you can get a great immune response in the brain without toxicity in the brain in certain situations. And what about glioblastoma, the tumor that is tougher to treat? And there was an interesting study out of the Toronto group that looked at the relationship of the number of mutations and response to nivolumab in a certain rare type of pediatric brain tumor that has very high level of mutations in the tumors. You can see thousands of mutations compared to, and the number of mutations is so much higher than even melanoma, where we know immunotherapy works well, and lung and colon cancer. So in this specific genetic brain tumor, they saw responses in the brain with actual shrinkage of the tumors over time. But there was consequent swelling, and patients had to be put on Avastin to control the swelling, get them over that part before they saw the response later, a pseudoprogression we call it. So I think there is opportunity for immunotherapy in the brain, even the stronger immunotherapies. We just have to understand who's going to respond, and then how do we manage the side effects. That's really the two key issues for immunotherapy. And what about non-glioblastomas, low-grade? There was a study done by one of my colleagues, Dr. Okada at UCSF now, who basically looked at peptide vaccines for low-grade tumors, which don't enhance on imaging. So this is, you can see the glioma here on T2 flare, but it doesn't take up the dye. It doesn't enhance like a glioblastoma. And when we give these peptide vaccines, he showed that you can see these, that the tumor now enhances a few months later. And this suggests that without any side effects. So in these low-grade tumors, you can get an immune response without tremendous swelling, without side effects. And then the immune response goes away later. We don't have information about what happens long-term, but in principle, you can induce an immune response to various types of brain tumors, and you can figure out the toxicities and manage the toxicities. And that's really what I think we're all trying to work on, is figuring out who's going to respond to what drugs, and how can we manage any potential side effects that arise. And that's really the next generation, what we call precision immunotherapy, is, precision immunotherapy, where we're picking immune approaches for specific subsets of patients and trying to really select patients for the next generation of precision medicine clinical trials based on immune characteristics. And again, this gets back to really what many people here try to do, which is translational research. You know, you need tissue, you need biomarkers, and you need to follow these patients longitudinally to understand what's happening to the tumors and how we can do a better job of selecting patients. So I'm going to step back a little bit and talk about new drug development, which I alluded to. So one of the things I think Peter will talk about later is that we all feel that the tumors respond and then become resistant later because there are stem cells that we're not targeting appropriately or adequately enough with the standard treatments of surgery, radiation, and chemotherapy. And these stem cells sit there, and they can progress immediately, or even months or years later, they can start growing. And this is one of the challenges, is because we're not really targeting those stem cells, and this is the reason why these tumors tend to come back. And so one of the factors that I worked with when in my postdoc at Dana-Farber was a protein called OLIC2, which is a transcription factor, which is very important for normal brain development. And it turns out that it's also very important to growth of glioblastoma. And there's a lot of data, but we published that shows that it drives tumorigenesis, it promotes resistance to radiation, it drives invasion, which is one of the big problems we have in surgery of trying to get it all out, is the tumors invade into the normal brain, so we can't surgically get all the tumor out. And these stem cells can also cause an immunosuppressive environment, so if we get rid of the stem cells, then the immune system may work better as well. So for many years, about 6 years ago, we actually targeted, using a lot of fancy computational chemistry at UCSD, targeted this transcription factor with small molecules and identified compounds that will inhibit that protein. And the idea is to use that molecule to combine with radiation and chemotherapy to get rid of those stem cells, with the hope that this will delay tumor recurrence or cure patients. And this is some of the basic science data that we published many years ago, where when we genetically knock down OLIC2 in glioma stem cells, you can see it slows the growth of tumors down, but it also makes them very sensitive to radiation, 2 gray, 4 gray, 8 gray. When you knock it down, they're very sensitive to radiation. So it's a good radiation sensitizer when we knock down OLIC2. And when we knock down OLIC2, we can also show in various other publications that it inhibits the invasion of these cells into the normal brain. So two good things that we're trying to address clinically all the time. And so that's what we did. We made a drug, and it's got all of the characteristics we want. It's an orally available drug. It gets across the blood-brain barrier, no clear cardiac or other side effects, and it's pretty easy to make. And we did studies in mice showing that the drug reduces the presence of OLIC2 positive cells, which is the brown staining, and quantified here. It also has an immunomodulatory effect that we are still trying to understand, but it takes away one of the immunosuppressive IL-17 cytokines. And when we treat animals with it, we can see improvement in survival of orthotopic mice, and with oral dosing of the drug. And when we combine with chemotherapy, there is a synergy in terms of improving survival as shown on this graph. There's also data with radiation where it also synergizes with radiation. So the plan is to really get this into human trials by the end of the year. The company's been working on this for, as I mentioned, 5-6 years, and they're ready to go to the FDA for a human trial by the end of the year. So that's one new drug, but I think, you know, we need to make more new drugs that are really specific to glioblastomas, and I think Dr. Dirks is going to probably talk about some of the specific pathways, and Dr. Baliga mentioned there's a lot of complexity and a lot of nodes or transcription factors which no one's targeted, which are critical for glioblastoma cell growth, but also similarly for other cancers, there are these nodes that no drugs are available for, that we can make drugs and open up new areas of drug development. And so one of the things that we're trying to do as part of Providence is to really put together a dream team, so to speak, of looking at all the heterogeneity and incorporating that information to select drugs that are going to benefit individual patients. And that's really the hope that we're all trying to do. And I'll leave you with this in the last few minutes, that you know, like I said, in practice, we have to do something for our patients while we still do the science and understand drugs. And there are a lot of tools available at our disposal. It's hard to get them. We have to fight with insurance companies, but there are tools. And this is an example of a patient that I just saw in the last few months, a 74-year-old with metastatic head and neck squamous cell carcinoma, okay? Not a brain tumor. But this patient had failed all the other, you know, radiation, a whole bunch of chemotherapies. And they came to us specifically for clinical trials. We looked at the tumor. We found out that the patient had a PD-L1 positive marker, which usually means you can respond to checkpoint inhibitors. He also had in the blood a marker of response to everolimus as well, which is that other drug I mentioned. So when we did the baseline scans, we saw all these, on the CT, the PET CT, we saw all these lesions. And you can see the dark lesions in the lung, the liver, gallbladder, and other areas of active tumor that's growing. And because I'm a neuro-oncologist, and when you have cancer spread from anywhere to the lung, it also means it's probably in the brain as well. We did an MRI of the brain, and he had 4 asymptomatic lesions in the brain, okay? No symptoms. So at the time we discussed, should we radiate the brain or not? Asymptomatic, maybe he'll respond to the immunotherapy. And we started nivolumab, ipilimumab, and everolimus. We were doing precision medicine with everolimus, and we were doing immune therapy with the nivolumab and ipilimumab. And surprisingly, or amazingly, 2 months later, all the lesions in the body shrank, and the PET scan looked better, reduced metabolism. And in the brain, everything shrank as well within 2 months. So this sort of highlights a new era that we're in of being able to have immune responses in the brain without there's no toxicity at all for these drugs, this combination. So we have to be a little bit more innovative, a little bit more creative, but we can get amazing responses in brain in certain subsets of patients that we don't 100% understand who can benefit from all these amazing drugs that are already approved, that are on the market right now. And that's really what we need to do, and I hope I've given you a flavor for what we can do in precision medicine and immune therapies, and what I hope in the next few years you'll see us integrating all this information to be able to make a difference for brain tumor patients. Okay, thank you.